Everyone. Thanks for joining today. This is Jason Key at SB Grid at Harvard Medical School in Boston. This is our first webinar of 2019. So thanks for joining. With that, it's my pleasure today to have Thomas Grant joining us from Hauptman-Woodward in Buffalo. So Thomas is the author of DENSS, and he's going to tell us today about electron density from solution scattering data. Go ahead,
Great.
Thomas.
Thanks, Jason, and thanks for inviting me out to give this webinar. So I'm going to talk real briefly, kind of divide this up into two sections, where the first part of this will be talking briefly about the algorithm itself and kind of some of the theory behind it, because it works a little bit differently than some other SAXS modeling algorithms. And then after the tutorial, to understand how it works, I'll go through a little bit of a tutorial on how to actually run DENSS on your own data. So just to give some quick background, a solution scattering experiment is a pretty simple experiment where you have a solution of particles. Rather than being in a crystal or on some kind of grid, you have a solution of particles that are exposed to an incident beam of X-rays, typically from a synchrotron or from a lab source. And because the particles are all randomly oriented, floating around in solution, they tumble, and they scatter the intensity in all directions. And so you end up with a scattering profile that's isotropic, and so you can radially average this profile on a 2D detector to give you a one-dimensional scattering profile of intensity as a function of Q, where Q is effectively the angle, what we call the momentum transfer. And in doing this, from this one-dimensional profile, we can get some basic information about the size and the shape of the profile of the particle. And so, for example, we can get information such as the radius of gyration of the particle or the maximum dimension of the particle. We can also get some shape information through something called a paired distance distribution function. But when it comes to extracting three-dimensional structural information from solution scattering data, there's been a variety of algorithms that have been developed. And so usually what these algorithms do is they define an envelope, and then inside of this envelope, they assume that the density is uniform. And this is a major restraint on the system, because at low resolution, which is what we're doing small angle scattering at, is at low resolution, you really can, for the most part, for relatively simple molecules, you can assume that particles are uniform density. They're pretty much just carbon and oxygen and nitrogen for the most part, and they all have about the same density throughout the envelope. One of the first ways that people tried to extract some kind of 3D structural information was just through simple shapes. You take something like an ellipsoid or a prism, and you fit the axial ratios, for example, of the particle, assuming that the interior of the envelope defined by this shape is some uniform homogeneous density, and then you can calculate the scattering profile from that and fit those parameters such that it best matches your data. Obviously, this is a very low resolution technique. It really just tells you if something is sort of prolate or elongated or oblate, things like that. Then there was a development for being able to combine a series of simple shapes, collections of shapes, where you could get slightly more complex objects. A big advance came when people started using spherical harmonics equations to define the envelope boundary. And once again, you assume that inside of this envelope, you have a uniform electron density. And this was really great because it allowed you to get much more complex shapes, but it was still pretty limited in terms of how complex of a shape you could get. You can't get things with cavities or things that really can't be represented well with spherical harmonics. And then a big advance came through the advent of bead modeling. And this is what most people associate ab initio modeling with when it comes to SAXS data, is bead models. And bead models are great because they allow you to define an almost arbitrarily complex shape with cavities or clefts and things like that, where each bead is the same, and you just assign them onto a hexagonal grid, and you generate thousands or even millions of different candidate sort of ensembles of beads or configurations of beads. And this, you then calculate the scattering profile from these configurations and pick the ones that best match your data. And then over the last decade or so has been a lot of effort in developing hybrid modeling where you can throw in some other information from things like crystallography or NMR to try and generate higher resolution models. So I would kind of put all of these under the heading of modeling. And what I mean by that is that in all of these cases, you're generating lots and lots of different candidate models, calculating the scattering profile, and then seeing how well it compares with your experimental data. And you do some minimization procedure like simulated annealing, for example, to try and define the best model. What I'm going to talk about is something I developed for actually calculating the electron density directly from the scattering profile. And this actually goes the other way. Rather than calculating scattering profiles from models and seeing which one fits best, we're actually here going to use the data itself to tell us what the density should be. And so it's pretty novel in that aspect. So how do we do that? How can we calculate a three-dimensional electron density map when we only have a one-dimensional scattering profile? Well, I'm going to kind of take you into single molecule imaging as a way of helping to understand this. And so in single molecule imaging, if you were to take the Fourier transform of a single molecule's electron density function, this is single molecule electron density function, or sorry, single molecule reciprocal space of lysozyme. So we actually take lysozyme, calculate the Fourier transform of the electron density, and this is what we get. And you notice that this is a continuous distribution of intensities in reciprocal space. It's a 3D space, and you notice that we have a very bright blob at the center. That's our small angle region. And then as we go out to wider angles, it's much dimmer, much fainter, less intense, and the fluctuations are continuous. And so the problem in single molecule imaging, theoretically, if you could do those experiments, is that if you get a set of intensity, these are intensities, if you get a 3D volume of reciprocal space intensities, you only have then information about the amplitudes. And what you really need are the structure factors. You need both the amplitudes and the phases at every point in reciprocal space in order to be able to do an inverse Fourier transform and calculate what the electron density is. So they have to solve the phase problem, right? So they know the 3D amplitudes from their experiment, but they don't know what the phases are. And so this is the phase problem. And so they've come up with some clever ways through iterative phase retrieval. And what iterative phase retrieval is the idea of sort of dividing the problem into two domains, where you have a real space domain and you have a reciprocal space domain. So the real space domain is where you have your electron density, and reciprocal space is where you have your structure factors, where the structure factors are complex. They have amplitudes and they have phases at every point in reciprocal space. And so these two domains are related by Fourier transforms, either a forward Fourier transform or an inverse Fourier transform. And the basic idea here is to take advantage of the fact that you have these continuous fluctuations in reciprocal space, because this provides oversampling and a little bit more information than you would get for example, from Bragg diffraction in crystallography. That's not oversampled, that's actually undersampled. And so the fact that we have continuous intensities in reciprocal space allows us to impose some new restraints on the system. And these restraints in real space are things like solvent flattening. Since we know the boundary of the particle, we can actually take a larger section, larger volume of space and flatten the region around it, since we know that solvent should generally be flat. We can also impose other real space restraints, things like positivity. We know electron density should be positive. And we also can impose some ideas of connectivity. We know that particles are connected objects. And in order to do this, we have to define what we call a support. And so a support is effectively the envelope boundary. And so there are different ways to do this, and I'll describe what I use in my case in a minute. But effectively what you do is you define a boundary of the particle, and that way you can apply solvent flattening outside of this boundary and things like positivity and connectivity inside of this envelope. And what you do is every time you iterate through this, you update your phases, and you get slightly better each time, and you can run this thousands of times until you get to some convergence of these phases. So the problem in solution scattering is a bit different, though. In solution scattering, because we have molecules tumbling in solution, rather than having this sort of 3D reciprocal space fluctuating profile, we actually have spherical averaging. And so the spherical averaging causes each region, each sort of Q shell in reciprocal space to be averaged. And so you end up with just a one-dimensional scattering profile, where it's this radial isotropic distribution. So what that means is that we no longer have three-dimensional intensities. So that means we don't have three-dimensional amplitudes. So rather than just having to solve the phase problem, we also have to solve what I call the structure factor problem, where we have to solve for both the amplitudes and the phases in reciprocal space. And so I developed an approach that I call iterative structure factor retrieval to do this. It's very similar to iterative phase retrieval. The first thing we do is we define a grid. And so this is a 3D grid. And so this 3D grid you can kind of think of as a 3D image, where it's composed of a bunch of pixels that we call voxels in 3 dimensions. And so at every grid point, you start off with a random density. So you take this 3D grid and you just assign random values to the grid points. And this is your initial random electron density map. From that, you calculate a forward Fourier transform, and that gives you a set of 3-dimensional structure factors in reciprocal space. And so those structure factors are complex. They include an amplitude and a phase. And from those structure factors, you can calculate the complex conjugate to come up with a set of reciprocal space intensities. So now these intensities, we need to be able to compare with our one-dimensional scattering profile. So we have to reduce this 3D intensity space to a 1D spherical average. And so the way we do that is by binning all of the grid points into a series of concentric Q shells, so that these Q values would match up with the Q range in your experimental data. And so by binning all of these grid points, we then can take the average of all the grid points that fall within each shell and come up with a one-dimensional spherical average of intensities as a function of Q. Now, when you compare this to the experimental density, of course, it's going to be wrong because we started with a random electron density. So the key is, how do we change the structure factors such that the intensities would have an average that matches our experimental data? So what we can do is we can actually calculate what the factor is to change all of these intensities by. So we change all the intensities by the same factor. We calculate the square root of the ratio of the experimental intensity to the calculated intensity, and that allows us to change all of the intensities in this spherical shell such that they have an average that matches your data. And we're actually going to calculate this scale factor as a square root of this ratio, and that's because we're going to change the amplitudes rather than just the intensities. So once we do that, we get a new set of structure factors in 3 dimensions in reciprocal space, and from that we can calculate an inverse Fourier transform and return to real space. And so this gives us some density, some kind of density that itself has a scattering profile that matches the data. Now the problem with this is that there are effectively an infinite number of different electron density maps that you can make that all have the same scattering profile, because it's a degenerate problem. And so this is where we apply these real space restraints, similarly to the way that they do it in single molecule imaging. So first we need to define a boundary for our particle in order to be able to do this. So the way we do this is through an algorithm called shrink wrap. And what shrink wrap does is it effectively blurs the density. If you just sort of imagine taking an image in Photoshop and applying a Gaussian blur, that's effectively what it's doing. It blurs everything so that neighboring points are similar, and then it defines the boundary as a region where most of the density is. Any grid points that have a density that's above some threshold, typically 20% of the maximum density in this blurred map, becomes your boundary. So this is now your support. The grid points in this region are now your support. And so now what we can do is we can flatten the density outside of this region, make sure that the density inside is positive, and we get some kind of cleaned up, flattened electron density map. Now that we've done this, however, we've changed the electron density map, so it no longer has a corresponding scattering profile that matches our experimental data. So we go through this cycle again. We calculate the forward Fourier transform, calculate the spherical average, rescale the structure factors, and recalculate a new density. And you go through this iterative cycle thousands of times until you obtain a final electron density map that matches both real space and reciprocal space restraints. And that's the basic idea. So this is a little animation of this process taking place. On the top, we have the random electron density to start with. On the bottom left, in the red dots, we have the calculated intensities, the calculated spherical average intensities from this current map up on the top. In the black dots, we have the raw experimental data. Now, rather than using the raw experimental data, what we actually use is a fit to the experimental data, and that's this black line. And the reason for that is because that takes advantage of the oversampling in the scattering profile that we get from our experiment. And so our target function is actually the black solid line. And on the bottom right, we have the chi-squared between the red dots and the black line. And so as I begin the animation, the first thing you notice is that it sort of immediately collapses to some kind of globular shape. And the reason for that is because it's gone from a random intensity to something that has some kind of particle-looking nature. And you notice these spikes, that's where shrink wrap is being updated. You can kind of see it chipping away at the volume as it goes here, as it converges. And you notice that at low resolution, once we dock in the structure, at low resolution it matches the overall shape of the expected structure relatively well. Now this is at low resolution. So typically something like 20 angstrom resolution or lower. Now, one thing to remember is that because we start with a random number, we're a random electron density, we're getting different reconstructions every time we run this. But what we found is that if you run this, say, 20 times, all of these reconstructions are effectively just perturbations about some low resolution electron density. So the overall envelope might change a little bit, the distribution of densities might change a little bit, but if you average them all, you can get some kind of low resolution electron density map. And so that's what we do, we average them. So you run this something like 20 times, and you align each of the different maps to each other map, and you average them to produce some kind of final low resolution electron density. Through the same procedure, we can get a rough estimate of resolution by taking these 20 maps and splitting them into two halves, even and odd, and aligning them and averaging them independently. And then what you can do is you can actually estimate resolution by looking at how well these two halves correlate with each other, very similar to how you do it in cryo-electron microscopy. And you can calculate a Fourier shell correlation, and that gives you some kind of rough estimate of resolution for the reconstruction. Now, this is really just sort of a proxy for resolution because you're not actually comparing it to a known structure. And so what I found was that if you simulate data from known structures where you know the answer, in those cases you can actually compare the final average with the actual structure. And what we find is that the Fourier correlation between the even and odd halves often reflects what that Fourier shell correlation between the average SAXS electron density map is versus the known structure. And so doing the even versus odd acts as a rough estimate of resolution to the actual particle shape. So there's some results from some proteins of varying sizes and shapes, and you can see that overall the electron density maps match the expected structure saw from crystallography. In some cases, it actually shows some additional regions. For example, in this one, there was something like 20% of the residues were missing in the structure, and they were present in the SAXS data. You can actually see where they are located. Similarly to number 10 here, a large chunk of 20 to 25% of the residues were missing. You can actually figure out how to align your particle based on where the terminal residue is, where those residues are attached. And gives you some rough idea at low resolution of where those extra residues are. So those were all cases with the proteins where in most cases you can actually assume a uniform electron density. It does a pretty good job. That's why things like bead modeling and have become so popular with SAXS, because they are generally a true assumption at low resolution. In some cases, however, that assumption breaks down, in particular for cases where you have large differences in the electron density at low resolution between different species in the sample. For example, this was a nice case that I found on a database online where you have this sort of dumbbell-shaped particle where it's a protein dimer that's connecting two micelles. And it's sort of a very complex shape to start with, which is typically difficult for SAXS to reproduce, these reconstructions to reproduce. And so additionally, this is a nice case of showing the different electron densities that can be found, because the protein has one electron density on average at low resolution. The micelles themselves have two different electron densities. They have the high density electron-rich head groups, and then they have the low density core where the lipid tails are inside the micelle. And so when you run DENSS on this, you can see these reconstructions where not only does it reproduce this overall dumbbell-like shape, but it actually is also showing you, if you look at a cross-section of the density, that the highest electron density regions are in the shells around each micelle, and you have low electron density regions in the interior and the protein region connecting the two. So it actually shows you the different density regions throughout the particle at low resolution. This is just showing that it's actually producing real electron density, just some simulations of simple spheres. So if you take a flat electron density as your actual target, it does reproduce that flat electron density. If you have a gradient where you have a low density in the center and a high density at the outside, it is able to reproduce that. If you do the opposite, where you take a high density in the center and a low density at the outside, it's also able to reproduce that. And finally, if you do something where you have two uniform regions where the one region is offset from the center, it doesn't do quite as good of a job, but you can see that it is showing the density offset from the center. Once again, these are all low resolution. It does have its limits, however. Particularly complex shapes are very difficult to reconstruct. Shapes that have a high aspect ratio. In this case, up at the top here, this aspect ratio is like a 10-to-1. It's like 400 angstroms across and 40 angstroms wide. So it's like a 10-to-1 aspect ratio. And you can see it did not do a particularly good job of reconstructing all 5 arms, but it got the rough shape and different regions. So it does have its limits. This is a nice case where you have this porin where these channels, you can actually see the low density channels in the trimer. And finally, this is just another complex shape to show that it can reconstruct relatively complex objects. This was a case that was a good example of the problem of enantiomer selection in SAXS. And so the problem in SAXS, or one problem in SAXS, is that different enantiomers or different mirror images of a particle will have the same exact scattering profile. And so when you run DENSS, you actually have to do some kind of enantiomer selection when you're doing the averaging, because otherwise you'll average out left and right-handed reconstructions. And in that case, you would end up getting some kind of average that was incorrect. And so we now have the ability in DENSS to actually flip each of the molecule, each of the reconstructions over all the different axes, and then average them such that they all have the same enantiomer. Now, it may not be that they have the correct enantiomer, but at least they have the same enantiomer. And you can see in this particular case that it did a good job of doing that. This is a case where if you took the left-handed reconstruction and averaged it, you would just average out to something that's then symmetric, which clearly this is not. This has a clear-handedness to it. I also chose this example because it's a very ambiguous shape. If you actually look at something called ambimeter, it's a way to score how complex a shape is. This is the shape that I found to be the most complex in the PDB that I could find, with a very high ambiguity score. Typically, you want something like 1 or less, or 1.5 or less, and this is on a log scale, so 3 is very high. And so not only is it able to reconstruct the envelope very well, but actually it is able to sort of see these interior domains at low resolution. You can kind of see these red hotspots for these 3 domains that pop up. Okay, so that's about it in terms of the sort of theory section of this, just so that you can understand when I go through the tutorial what the different parameters mean and why you'd want to be able to modify them and how to modify them. There's some things that I'm working on right now to help improve this, like throwing in known partial structure. Actually, this non-crystallographic symmetry is now available in DENSS. If you have something where you know it's a dimer, for example, you can apply P2 symmetry, and it will do NCS averaging to improve that. And it turns out to be really powerful. If you go back to this guy here, so this is without any symmetry. If you apply fivefold symmetry to this, it does a very good job of actually reconstructing the sort of little ring in the center, and then these 5 arms on either side. And so it does a very good job to impose the extra symmetry. And then there's a series of other features that we hope to be able to add. Okay, let me see. How am I doing on time? Okay.
Before you jump in, I had a question. Maybe now's a good time to pass it on.
Sure.
A question for the structure alignments, presumably for the averaging section of the algorithm. How are you doing the alignment between the different the maps, the densities?
Yeah, so that's a good question. So there's 2 approaches, and I'll actually show this in a second when I go through the tutorial. But there's 2 approaches. So the original one is through a software package called eMan2, which is a very popular package for cryo-electron microscopy. So a lot of the type of maps that we get out of DENSS are low resolution maps, and so there's a lot of cryo-EM tools that are useful. We actually do everything in terms of MRC files, which are common cryo-EM map format. And so we use the tools available in eMan2 to do this, where effectively what happens is there's a correlation that's calculated between, you take map A and map B, and you move them and you minimize the alignment between the 2 maps such that you would maximize their correlation. So you effectively multiply the map by, map A times map B, and you normalize it, and you look at what the highest correlation you can get is. That's effectively what it's doing, and it's looking at translation and the 3 Euler angles in order to optimize the rotation. And then you do that for each of the 20 maps. So you could theoretically do a full pair calculation where you're aligning every map to every other map. But for efficiency's sake, like if you have 20 of them, that's 400 calculations. So that would take a long time. So for efficiency's sake, we use a sort of binary averaging procedure. It's sort of this binary tree kind of thing where you pair them off. If you've got 20 maps, you pair them into 10 pairs, or I guess 8 pairs, and then you average those 2 in each pair, and then you average those pairs until you converge to 1 left, and that gives you some kind of reference, and then you average all 20 to that reference, or actually you split them up and you average them to 10, so you have 2 independent alignments. And so that's effectively how it's done in eMan2. We do something very similar. We now have a built-in averaging procedure inside of DENSS. And so it does something very similar to what eMan2 does. And that's just so that we can have something self-contained in case people don't have access to eMan2 for it. So that's about it for the theory part of this. Were there any other questions before I go on?
There was one more, but I think we'll save it to the end.
Okay. So the software is called DENSS, which stands for Density from Solution Scattering. It's all open source, written in Python. It's available here for download. The main website for DENSS is tdgrant.com/DENSS, and this has a lot of useful, tutorial, for example. It's got a lot of tips, how to install it. I mean, this is SB Grid, so you guys should have all this installed for you. And it gives you a lot of information how to actually run DENSS and what the different parameters mean. And so I'll show you that in just a second. DENSS is now actually integrated into software called BioXTAS Raw, which is a popular GUI application for reducing and modeling data. It incorporates a lot of different modeling suites to be able to do some modeling with a GUI, in case you like GUI, because DENSS is pretty much entirely a command line driven program. And so Raw is actually it builds DENSS right into it, so you can do it with a GUI. We have a web server that's available for some simple reconstructions, and that's this guy here. It's called DenseWeb. just dense.ccr.buffalo.edu, and this will run the whole pipeline where you upload your scattering profile, and it will do 20 reconstructions of DENSS, and then it will do the alignment and averaging and everything. Right now, it's in sort of a beta simple version mode, where it's in what we call fast mode, so it just does a simple low resolution reconstruction to give you a quick idea if you've got a simple shape. If you have more complex shapes, you want to run it in slow mode, or even some kind of optimized slower mode, which I'll show you in a second. And that's not quite available in DenseWeb, but we're updating it currently, and hopefully soon it'll be able to do those more standard slow mode reconstructions. And so this is the website, and I'll go through this real quick, and the tutorial that we'll go through will effectively be a real quick version of what we're going to see in this website. So you can either go to dense.org or tdgrant.com/dense. Dense.org will just take you to tdgrant.com/dense. So we'll click that and load up the website. Well, it's already up here. So this is the main website, and so you can get some information about DENSS, about downloading DENSS, about how to install DENSS if perhaps you want to install it on your own. This is sort of just a quick way of doing this. We recommend something like Package Manager, like Anaconda, if you'd like, for installing all the packages required. It really just requires NumPy and SciPy, which are two very common Python packages. You can also have eman2 installed if you want to run the eman2 averaging, and it gives you a quick description of how to install both eman2 and how to install DENSS, and if you want to just update it. I'm updating DENSS pretty often. Like there's often a lot of bug fixes, so always try and keep DENSS updated as often as you can. Okay, so over here on the menu you see tutorial, and so this is what we're going to go through. If you just click on the tutorial link, it'll give you a list of the different tutorial pages and some quick background that we just went over. Pretty much at every page, at the bottom of every page, there's a form for you to contact me. If you have any questions about anything, please feel free to contact me, and I'll do my best to fix bugs that you find, or include some features that you like, things like that. So the first thing we'll do is we'll go to the introduction. All right, so here you see a list of the scripts that DENSS has included. It's actually a suite of scripts. The main script is something called dense.py. And so pretty much all of the scripts, well, at least most of the scripts, work through sort of this idea of dense.something.py in order to make it easy for you to find the scripts. So dense.py is the primary one that'll just run a single reconstruction of DENSS when you give it the scattering profile. Dense.all.py will do the full pipeline of running 20 reconstructions, aligning them, averaging them, doing the enantiomer generation. Same thing for superdense. Superdense is a bash script, however, so I'm not sure if it'll run on Windows, but that's one of the reasons we created dense.all.py to make it easy for people using Windows. But superdense will run the eman2 version of the averaging. And then there's a series of other scripts here that you can look into if you're interested, for things like aligning a PDB to a map, or refining a map, for example, if you're interested in that, or getting various information from structures and things like that. Okay, so the first thing to do is to get some data. If you're interested in just testing it out, there is some example data provided with the download of DENSS in the GitHub repo. It's just simulated data from lysozyme structure, and that's what we use, just a simple way of getting something small and globular so that you can see how it works. And so one of the things to note is that we don't actually give DENSS raw data. If you remember from what I mentioned a few minutes ago, we actually give DENSS a smooth fit to the data as sort of our target function. There's a few ways you can do this. Effectively, DENSS just accepts .dat files generally. So if you have some way of calculating a smooth fit to a curve that you'd like to use, just make sure what you provide to DENSS is a .dat file. So these .dat files are just ASCII text files that are given as 3 columns, Q, where Q is the momentum transfer. It's important to note that the momentum transfer here is given in inverse angstroms. So if a lot of beamlines give you something that is inverse nanometers, you just want to go and convert that to inverse angstroms, which means just divide by 10 in the X column. And then the second column is the intensities, and the third column is the error on the intensities. And so that's just a simple ASCII text file. So if you don't have some way of smoothing your data, you can use, there's one built into DENSS that's called dense.fitdata.py. And I won't show you that because you can read about it and you can test it out yourself. But this is a quick little image here showing how it works. All you would type is dense.fitdata.py-f is to tell you that this next parameter is the file name for your raw data. And this is your raw experimental data. And what it'll do is it'll open up this little widget here. This is all done in matplotlib. So you got to have matplotlib, I believe, version 2.2 or later, something like that. And it'll open up this little interactive widget. And so it'll plot your data. It'll give you an initial fit. And then on the right, it'll show you P of R. Now, what's important here for DENSS is not P of R. We're not particularly interested in P of R. We're interested in this red line. We want this fit to be nice, a nice fit to the data. And so there's some parameters here like Dmax. You can get an estimate of Dmax by looking at your P of R curve. It'll start off by default as at 100. And so you can just drag this little slider back and forth to do larger or smaller Dmaxes. And there's also this smoothing parameter alpha. If you're interested, like a lot of times this will have very a lot of ripples in it, like very large ripples from the Fourier truncation. And so you can use this alpha parameter to smooth it out, give you an estimate. But again, if you over smooth it, the fit will get worse. And so you want to make sure that your fit is best. And that's what's important. So once you have that, you can save the file. And that'll create a file called, in this case, if you were to load in like 6lyz.dat, it would output 6lyz_fit.dat. And I'll show you that in just a second. The other option, which is a very, very popular option, is to use a program called Genome from the SAXS suite. So SAXS is a collection of SAXS programs that's very useful. It's really the most popular for biological SAXS. And Genome is a program that's built in for doing this, effectively the same thing, just with some different algorithms. And so you can use this within their Primus GUI. It's got a nice little GUI to do this, and calculate, again, a fit to the scattering data. And from that, it'll save something called a .out file. It'll have a .out extension, and you can use that as input into DENSS. If you're interested in doing that, I would just recommend you read this little section down here. All right, so now we'll go to actually running DENSS. So now that we've kind of prepped our data, so there's you can run DENSS directly with the .out file, or you can run it with the .dat file. And if you run it with the .dat file, you have to also give it this Dmax parameter. So remember, the way that DENSS works is it first creates a grid. And in order to make sure that the grid is of an appropriate size, you have to tell it how big your particle is, at least a rough estimate. So in this case, all Dmax is used for is defining the box size. And so it does this by multiplying D, this maximum dimension, times an oversampling ratio, which by default is 3. You need a minimum oversampling of 2 to make the phase retrieval work. And so I do 3 just to give it a little bit of leeway in case your Dmax maybe were underestimated or something like that. And so by default, it uses 3. And so it multiplies 3 times 50 to give you an initial edge length of 150 angstroms. Now, D, if you don't give it D by default when you use a .dat file, it'll assume it's a Dmax of 100. But that's generally not accurate or not good enough for most, or maybe not for a lot of particle shapes. If you use a .out file, however, it'll actually extract the Dmax from the .out file. It'll parse this file and extract the Dmax from the .out file. So you don't need to give it a Dmax if you give it a .out file. Okay. So DENSS works in sort of a few different modes. So for convenience sake, I now have these fast and slow modes. So the default is slow. And effectively the difference that between fast and slow, or what defines how fast DENSS runs is the sampling ratio. Or sorry, not the sampling ratio, but the sampling. The sampling is effectively how many grid points within each dimension of the box you're going to have. So by default, in slow mode, it's 64, which means if you have a box length of 150 angstroms, for example, for our lysozyme case, in slow mode, it will divide that up into 64 voxels in each of the 3 dimensions. And so you'd end up with a voxel size of something like 2.4 angstroms, whatever 150 divided by 64 is. And so because of that, it's 64 cubed, so you have, however many voxels that is. And so the speed goes not quite, but almost as the cube of the size number of samples. It's actually n log n cubed, or whatever that Fourier transform equation would be. So that way you can actually change the speed of the program. If you want something that's faster, you can run it at a lower sampling, like 32, for example, with fast mode. And what that does is it makes it so that you have larger voxels, and so there's fewer of them, so it runs a lot faster. However, the problem is now that their voxels are larger, you might not be able to contour particularly complex shapes. So if you have something that's simple like lysozyme, 32 might be okay, but if you have something a little bit more complicated, you really want to stick with 64, or even more in some particularly complex shapes. Particularly if you have large aspect ratios, a lot of times for that you need a high sampling ratio in order to make it. I'm sorry, not a sampling ratio, a high sampling in order to make it be able to reconstruct those complex shapes. So these are some of the options that you can have. So the oversampling by default is 3. In some cases, if the fits aren't particularly good, you want to raise this to like 5 or something like that. And I'll show you that in a second. -n is the number of samples that I was just talking about. By default, it's 64. You can also set the number of electrons with this option. And by default, it uses 10,000. But if you want to get something where you have the electron density is absolutely scaled in terms of electrons per angstrom cubed, you actually have to tell it what the number of electrons in the object is. And then all it does is it scales the intensity to the number of electrons squared. I mentioned that we recently included an option to have symmetry. So if you have a dimer, you would type -NCS2. And so the way that works is by first aligning the principal axes of the map to the XYZ axes. And so when you calculate principal axes, there will be a largest axis, sort of a middle axis, and a smallest axis. And so by default, it'll align the largest axis to X. And so then it'll do the symmetry around the largest axis, or around X. However, if you know something about your particle, maybe you know that the largest axis isn't the symmetry axis, maybe it's the smallest one. For example, in that sort of snowflake 5 pentamer kind of thing that I was showing you a second ago, it's actually the shortest axis that is the axis of symmetry. And so you could tell it do the NCS around axis 3 in that case. And so this just shows you how to do that. All right, and then finally you can change what the output file prefix is, and also this -m is where you change the mode, either fast or slow. But then there's a whole host of options. All right, so why don't we go ahead and test this out. So I've kind of got a little thing set up here. I've got my original data here, which is the simulated data. I've got this 6lyz.out file, which is the genome file that I mentioned you can create. I'll try and get this done real quick. And then there's this fit file. Just for simplicity and for trying to speed this up a bit, I'll just run with the genome file. So if you just type dense.py-f, which -f, I'll show you real quick all the options. You can get all the options of any of these programs by typing -h, and it'll give you a list of all the options. And there's a lot of them. The ones that I mentioned to you just a second ago are all up here at the top. But -f is the main one to give you the data file. And then if you need to provide a dmax, -d, and then these other options. But there's a whole host of options here if you want to edit how shrink wrap works or things like that. There's a lot of options for you to be able to do that. So we'll just get this running and I'll explain what it's doing. Okay, so as it runs, it prints this output to the screen as it's going through the different iterations. So on the left, we have the step number, so it's done 500 steps now. The second column is chi-squared. That's the estimate of the discrepancy between the current electron density map and the experimental data. Radius of gyration here is calculated directly from the electron density map. One of the things to note about radius of gyration is a lot of times at the beginning of the reconstruction process, these could be, some nonsensical numbers. It might start off with a negative value or something really large or something like that. And that's really just because with the Fourier transform, there's periodic boundaries and the RG calculation doesn't really understand when something floats over a side. But it recenters things and it gets more globular as it goes on. So it will converge to something more reasonable. And the support volume is the volume of the voxels defined by the support. And so for the first 5,000 steps, the support doesn't change. And the only thing, it's just the entire grid is the support. So it's not doing any solvent flattening to start with. And the reason for that is we can actually just apply positivity. We can just make sure there's no negative density. And what that allows it to do is without chipping away at anything that might chop off parts of the molecule, it allows it to come up with something globular-like using just that one restraint. And now that it's past step 5,000, it's starting to update the support, and you can see this sort of chipping away as time goes on. All right, so as it goes, it will create this 6LYZ underscore current.mrc, which is just telling you what the current map is. If you double click it and you've got Chimera installed or PyMol or something, it'll show up. So this is just the current electron density map that we're starting with. Now, one of the things you notice, there's this weird density on the outside, and that's just because by default, Chimera will bring the threshold all the way down to almost 0, which there's some noise out there. So if you just increase it even the tiniest bit, it goes away and it shows you what your electron density map is. I'm not going to go into using Chimera, but just for a couple tools that are useful, everything is in if you go Tools, Volume Data, there's a lot of useful little widgets in here, like the Volume Viewer is this thing up here, and that's how you control like the threshold, for example. So this value here is what the electron density is. You can see that as you raise it or lower it, you get more or less density being shown. And so you might wonder, well, how do I know what a good density or a good threshold is? You get some kind of estimate of your particle, which a lot of times you can get from SAXS data, particle volume, sorry, that you can get from SAXS data. In the case of lysozyme, it's something like 20 angstroms or 20,000 angstroms cubed. If you go over to Tools, Volume Data, and then down to Measure Volume and Area, that's what this little window is. If you change the threshold, you notice that number for volume changes. And so you bring this up to where it says, say, 20,000. And so this is about what your particle, envelope should look like. All right, so that's just a basic quick tutorial of how to use Chimera to view your map. And so that'll update about every 100 steps. And that way you get an idea of what's currently going on as it's running, in case you want to take a peek. So these are all the output files that it creates. It creates a lot of output files. The main one is 6lyz.mrc, if that's what you called your prefix. By default, it just uses the data file name. And you can just open that up in Chimera, just like we just said, and do exactly what we just did, right? And then it gives you the same map for what the support is, what that envelope is. Statistics by step is literally just all of these statistics that it's printing to the screen. It just records it in this file in case you wanted to plot it. And then this file, this map.fit file, it gives you the fit. And so it actually will plot these for you if you got matplotlib installed. And so, for example, this is the fit. And so the black line in this fit is the supplied data, the smooth data that you gave it. And then the blue circles are the interpolated data. And so what that means is we actually have to take the black line and interpolate it to the Q values that are actually going to be used for restraining the intensities. And so that's the blue circles. And then the red circles are what the actual final scattering intensity values were. And you can see that it fits very well. And here on the bottom, we have the residuals, just to give you an idea. This region over here is actually used to extrapolate the data. And so one thing is that when you have a box that has, voxel size, in the case of lysozyme, I think it's like in this particular case, the voxel size was like 2.4 angstroms or something like that. But our data doesn't go out to 2.4 angstrom resolution. And so it has to put something there. And if you let it just go randomly and just do what it wants, it'll oftentimes give you some artifacts that are not realistic. And so we just use Perot's law to extrapolate it in not a particularly realistic way, but these are all really high resolution terms that don't have a huge impact. But if you don't restrain them, it can do some funny things. So that's all that is. Okay, so we can also look at all of the statistics as a function of how the reconstruction proceeded. And this is chi-squared, the fit of the scattering profile. And you can see that, it converges, or it decreases monotonically for a while, and then around step 5,000, it immediately shows a discontinuity, and that's because that's where shrink wrap started. So it didn't allow for noise in the solvent region anymore, and so it shoots up. Sometimes it actually goes down. But then you can see it's iterating more and more as shrink wrap is updated and converging to a solution, and eventually it stops at step 10,000. Sometimes it'll stop earlier if it actually finds that the chi-squared converge sooner than step 10,000. So you can also look at, for example, RG, and this shows you how RG changed as a function of the step number. A lot of times you'll see these sort of discontinuities, and the reason for that is either because shrink wrap was started, or because about every 500 or 1,000 steps, it recenters the object, which helps to make the RG calculation a little bit more accurate. And then finally, we have the support volume. Importantly for the support volume is it's the volume of the voxels that contain the object. So it should not be construed as the volume of the object. It will always be larger than the volume of the object, but it gives you kind of an upper limit for what the volume of the object is. Okay, and so that's for running a single reconstruction. It's just as simple to run a full reconstruction, where you're actually doing the full 20 of them, and average and align and do an enantiomer selection, and things like that. And rather than DENSS.py, which is for a single reconstruction, you can run just DENSS.all.py, and that will do the built-in averaging procedure in exactly the same way. If you find that you added a bunch of the parameters for DENSS.py to get better reconstructions, I kind of recommend that you run a single one, make sure it fits, update parameters however you need to make it fit for one, and then you run the total averaging procedure. You can give it the same options. It accepts all the same options. It also accepts a few extra options, and those extra options are, if we scroll all the way to the top here, are, for example, the number of maps. And so by default, it's 20, but you can theoretically have more. And particularly complex shapes, you might need more in order to be able to sample enough of the configuration space. You can also run it in parallel if you've got multiple cores. By default, it just runs it on a single core. And if for whatever reason you would like to, you can turn enantiomers on and off. And so you can run that. I won't run it now. It takes something in the neighborhood, depending on the number of cores you have and how you're running the system, like in terms of the parameters, something like 1 to 2 hours on a good system with a lot of cores. And so don't be surprised if, on a laptop where it's like dual core or quad core, if it takes 4 or 5 hours, particularly for the enantiomer selection. That's really what takes most of the time. So I've run one for you here, so that you don't have to wait. It generates all those files, so you can actually see all the individual reconstructions. It will also align all of them. It'll actually also save all the aligned reconstructions. And then it'll give you the final average. So this is the final average from the whole thing. And if you want to do some certain simple things, you can do things like alignment right inside of Chimera. So this is from 6LYZ of a PDB file, for example. If we open up our model panel, we can rotate just one object give it some kind of rough alignment. We can make our map transparent actually so that we can see the PDB inside of it. Give it some kind of rough initial estimate. We can change our volume so that it's around 20,000. And then you can use this fit and map tool, again in the tools, volume data, fit and map, to actually do a quick fit of, to do a quick fit of the structure to the map. Okay, so that's the basic idea in Chimera. You can actually also load this up in PyMol if you like, if you're more of a PyMol fan. The advantage of PyMol, Chimera is really nice for doing a lot of these functional things. PyMol is really nice for actually viewing or making publication quality images. You can actually load that map in and use this volume tool, this volume display. It's really nice actually. So by default, it just does one single envelope at a single density point. But you can actually make it like nice and colorful and show how the density changes inside the particle, which is really nice to be able to see that. And you can use this volume panel. Sorry, I kind of went fast there. Coloring and then click panel, and it pops up with this guy. And again, I won't go through all the things that PyMol can do, but you can see like all the density inside your object and how it changes. And so that's the basic idea there. And real quick, I'll just go through the log file. So if you want to, it also saves a log file with all the information, what version of DENSS, what files you gave it, what parameters you gave it, what the final real space voxel size is, for example, and the final pieces of information, statistics that it got from it. And this random seed, if you wanted to, you could use this file, take all those parameters and put them into DENSS and get exactly the same thing if you gave it the same random seed. So that's that. One thing to note is that if you were to do the full averaging procedure, this final average map is unlikely to have a scattering profile that actually matches your data because it's an average of a bunch of different reconstructions. And so there's now an option in DENSS. It's one of these scripts, dense.refine.py. You give it the same file, and then you can give it that averaged map, and it'll actually take that. It'll actually take that map, and it'll refine it. So it'll start with that as your seed rather than using a random number. It'll use that map as your seed, and it'll start there. And that way, you get sort of the benefits of an averaged map, and you can refine it against the data so that it also matches the experimental data. And I tried to go through that as quick as I could. There was a lot to go through, but I think with that, I will finish up the slides here. Here's a quick plug for our new book. If you're interested, there's some details about this. This is sort of an unconventional book. It really is looking more at intuitively understanding the connections between theoretical and practical applications of SAXS in biological SAXS. And so finally, I would just like to acknowledge a series of people that have helped in terms of a lot of discussion to help make this work. So with that, I think I am done. And if you have any questions, I am happy to answer them.
I had a couple of questions. I guess one, just thoughts, not even questions really, but I was thinking about orienting the models within these envelopes, and I was wondering about, if you've got a cofactor, you happen to have some, heavy atoms or some selenomethionine protein lying around that you happen to use. Is that something you've looked at? Is it advantageous? Does it make a difference?
So at this stage, no. I haven't looked enough into it to know for sure. My anticipation would be that it's not going to help a whole lot, and the reason is just because it's too low resolution. The primary limitation of this is just the number of pieces of information you have to reconstruct an object. And so it's going to be low resolution. So one of the questions that I get asked a lot is, if you give it wide-angle X-ray scattering data, do you get higher resolution than if you give it just SAXS? And the answer is generally no. And the reason for that is because as you go to higher resolution, the number of pieces of information that you need to reconstruct the object at high resolution goes up as the cube of the resolution. Whereas for SAXS, the number of pieces of information you get from the data only goes up linearly, and so you lose out on 1 over n squared when you're actually reconstructing it at higher and higher resolution. And so you're still fundamentally limited, even with high resolution data, where things like heavy atoms would actually show up. You're still fundamentally limited to low resolution reconstructions just because there's not a whole lot of information. So if you have something like heavy atoms in there, it's very unlikely they're going to show up just because they don't have at low resolution enough of a density difference relative to the other sort of low resolution regions. Whereas if you had something that was a large collection, like a gold nanoparticle attached DNA or something, that'll show up. But some single atom like that generally won't show up. Theoretically, you could adjust the algorithm to be able to deal with anomalous signals. Like adjust your Fourier transform so that it doesn't require a real transform. It can be complex in real space. Then you could theoretically adjust that to help with that. I haven't done it. I don't know if it would actually end up helping a whole lot, but in theory it could.
One other thought that came to mind when I saw your envelopes that had crystallographically determined models that were missing domains or termini, or that this would be a phenomenal complement to crystallographic structure determinations for even molecules that are, let's say large complexes at, 3-ish angstroms, and, people are trying to find the domains and they're not even totally sure that they're in the crystal or that they're, so they would be able to take all of their images and say, produce some sort of spherically averaged scattering data and then somehow devise an envelope that they could see, like, oh, I've got a domain over here.
Yeah, theoretically, sure. If you could, sort of separate out the biological portion of the crystal structure of the density in the crystal, separate out from what the I guess the asymmetric unit, what the biological unit is really, and separate that out. Yeah, you could theoretically do something like that.
Yeah, it would be a very useful tool, I think, for more and more of these huge complexes or
And one of the things that I'm working on now is making it so that you can actually so there's a tool in DENSS now that you can calculate what the density from a structure is. It'll use a Fourier transform and attempt to sort of clean up all the truncation ripples to calculate what the density is from a structure that actually matches your SAXS data. Because typically, if you calculate the density from a structure with normal Gaussian approximations, they don't look like what the scattering profile is. But if you use a Fourier transform, it does. And in doing so, you could then use that as like a restraint in your system, which is what I'm working on now, to be able to build what the rest of this actually looks like at higher resolution. Rather than building the whole thing ab initio, you could just restrain this and build this part ab initio to try and get a better idea of either complexes or missing domains, things like that.
Oh, that's great. All right, well, with that, Thomas, thank you very much. That's really exciting stuff, and I'm excited to see where it goes from here.
Yeah, thanks for having me.
And everyone else, thank you for joining, and take care.